And so now I will introduce our first speaker, Reid Selby, who will be presenting their work on developmental transitions in lettuce.
Hey everyone, thanks for coming and thanks for the opportunity to speak. Um, let's go ahead and get into it. Okay, is that coming through okay? Sweet. All right. Yeah, thanks everyone for being here. Um, like Eric said, I'm a graduate student at Auburn University, and I primarily study stem cell dynamics and development in the Asteraceae, and we found some um, pretty unique stem cell dynamics within the Asteraceae. I'm excited to share with each of you today. Um, but to give you some initial context to our work, I'm part of a larger group of researchers spanning a few different labs. We have our lab led by Daniel Jones here at Auburn University, Dr. Jennifer Mandel at the University of Memphis, John Burke at UGA, and then we do some collaborations too with Mauricio Bonifacino as well as Zach Nimchuk's group at UNC Chapel Hill. Um, our primary big difficult question that we're sort of, well, one of them that we're looking at as a group is sort of where did the capitulum come from? It's a defining characteristic of the family, but we don't really understand its evolutionary origins very well. And to get at this question, we leverage a combination of comparative genomics, comparative developmental transcriptomics, and then what I particularly specialize in is functional validation of some of these findings. So to give you kind of an outline of what we're going to go over today, we're going to first start by talking about what a capitulum is. This might be surprising. We all know what a capitulum is, especially this crowd, but there's a lot less known than you might think. The broader underlying question behind this is how do novel forms like the capitulum arise? And then the two hypotheses we'll compare and contrast are essentially did the capitulum come from an entire inflorescence or did it evolve from a singular flower, which might sound kind of strange. Next, we'll discuss some of the work I'm doing to understand what genes might be regulating capitulum development. I use lettuce as a model system for this type of work. And then finally, I'll share some results looking at clavata signaling within lettuce that teaches us about Asteraceae development a little bit more broadly. And I'll end on sort of a crazy out there idea, looking at how we might be able to use root meristems to understand the capitulum as a whole. So what is a capitulum? We all know that a capitulum is a compact inflorescence in which many individual florets are all borne on a singular receptacle that ecologically functions as a singular flower or a pseudanthium. This is widely thought to be one of the reasons that this family is so successful. While the conventional hypothesis for the origin of the capitulum is that it evolved from a thyrsoid inflorescence or a highly condensed raceme. There isn't actually a lot of direct evidence suggesting this, and while it's a less popular, less common theory, some researchers do think that it's possible the capitulum actually evolved from a singular floral meristem. This is called the floral unit meristem hypothesis. But before we can get into the sort of nuanced differences between those two potential evolutionary origins of the capitulum, let's step back and look at plant development a little more broadly. As most of you probably know, plants grow from cell divisions within the meristems. And meristems are formed in the embryo and are maintained throughout the entire life of the plant, and they house undifferentiated stem cells. These stem cells will continue to divide throughout the entire life of the plant. There we go. will continue to divide throughout the entire life of the plant, producing new organs. Upon stem cell dividing, one of the daughter cells typically retains its stem cell identity, thus replenishing the stem cell pool in the meristem, while the other differentiates. And this process of differentiation is what leads to the organs that we see plants produce, like leaves, throughout their entire lives. Because this process of differentiating and renewing stem cells is so crucial to plant development, the pathways regulating this process are usually highly, highly conserved, because if something goes wrong, you're not going to develop properly. But while they're highly conserved, we do see variation in these pathways, particularly occurring during states of developmental transition, such as the shift from vegetative to reproductive growth. But we also see um, crucial variation in these developmental pathways between species. So while these pathways are broadly conserved across all plants, there are some key differences that we observe that kind of underlie the morphological diversity that we see in the plant kingdom. So where did this idea come from, that the capitulum might have evolved from a singular flower? Basically, the initial observation was that stem cell consumption as capitula develop sort of superficially resembles floral meristems. So here we have a diagram of a developing Arabidopsis inflorescence meristem. In the center, you have stem cells dividing to produce the inflorescence itself. And then all of these sort of numbered pockets you see on the outside there are flowers that are at different stages of development. Those colored rings you see are the floral whorls beginning to specify there. As an Arabidopsis inflorescence develops, you see sort of a budding, almost a splitting of these primordia off of the central inflorescence meristem, kind of one at a time as the inflorescence develops. However, if you look at a capitulum as it develops, you see a pretty rapid expansion of the stem cell niche marked in purple here. And that continues to get larger and larger and larger. And instead of flowers budding off one at a time, there's sort of a pause on any flowers appearing. And then they will sort of rapidly all appear all together in an acropoidal manner. Here's another look at some capitula meristem as they're developing. And you can see at this stage here, what might be called a floral unit meristem. You have a broad, naked meristem without any clear floral meristems forming, and then rapidly all at once you start to see floral meristems budding off in the side there. There is some molecular evidence that this might be the case, that a capitulum might genetically more resemble a singular flower. For example, if you look at the gene expression or the expression patterns of the gene leafy in Arabidopsis, we've known for a long time, this figure's from 1997, that floral meristems are sort of marked by the expression of this gene, not the inflorescence meristem. But if you look at a developing Gerbera capitulum, you can see this leafy gene, which typically marks floral meristems, is expressed across the whole thing. Now, while this is interesting and is kind of a catchy, unique idea, what I think is probably a bit more likely is that the capitulum is actually derived from a thirst-like inflorescence instead. And there's been some really cool work looking at inflorescence development across this MGCA clade here, particularly in the Caliceraceae here, showing that there is probably more evidence for an inflorescence-derived origin of the capitulum here. So did the capitulum evolve from a singular flower? All the evidence presented so far, and there's not too much, can sort of be interpreted either way. And to get to the bottom of this question, we really do have to start producing a lot more data. And our group has been brainstorming different ways that we can start answering this question. So as you kind of saw earlier, the Asteraceae, particularly the these 4 or 5 families here encompass many different inflorescence types, ranging from solitary flowers all the way to the capitulum we see in the Asteraceae. And while each of these inflorescence types might look drastically different from one another in their final product, they all undergo similar developmental transitions and they pass through similar developmental states that are roughly homologous to one another. We can leverage these roughly homologous states of meristem transitions to do some interesting comparative transcriptomics to understand how gene regulatory networks governing development in each of these species might differ or might be similar. So let's take another look at this developing sunflower capitulum. At 10 days here, we have a vegetative meristem, which is fairly small and flat, and it's producing leaves you can see budding off here and here. At 20 days, you can see the meristem starts to expand a little bit due to an increase in the number of available stem cells. And then at 30 days, at this inflorescence meristem stage, you see a pretty drastic expansion. The stem cell niche is fairly large, and there's a significant doming of the meristem. However, you don't see any floral primordia begin to form until here. And in the outer edges here, you'll get ray florets beginning to initiate, and then towards the center, you can see your disc floret primordia forming. So looking at this sort of developmental transition state, we're asking the question, is this stage here developmentally more like an inflorescence meristem or more like a floral meristem or like a floral unit meristem? So you can see there's a lot of potential for maybe interesting gene discovery looking across all of these different species and comparing them to one another. But gene identification is really just the first step in this whole process. We really need a system for functionally validating anything we discover. That's what's been lacking in our understanding of capitulum development thus far. And so to do that, we have assembled a genome for a cultivar of lettuce that we've been using as a model system. This lettuce cultivar is easily transformed via shoot regeneration and tissue culture here. It expresses transgenes fairly readily, as you can see from this 35S promoter driving GFP expression. It's a rapidly bolting variety, which is nice if you're trying to use it as a model system, gets those generation times down, and it also produces a large abundance of capitula, which is a unique advantage to an organism if you're using it to study capitula specifically. So just some statistics on our reference genome we assembled for this cultivar. We use a combination of PacBio Revio long reads and OmniC chromatin conformation to assemble a haplotype resolved genome. It looks like we've got just about the right size for the genome there, and we've got a clear 9 chromosomes within each haplotype, as well as some pretty decent BOOSCO scores, indicating we're on the right track there. Just some quick comparisons to other varieties of lettuce. There are genomes of different lettuce varieties, such as Salinas or stem lettuce here. And if we look at structural variations across these cultivars, we actually see a high degree of synteny and a low amount of structural variation between each assembly, serving as further quality control. All right, now that we have a reference genome, we can turn to transcriptomics across each of these stages to better understand how capitulum development is happening from a gene expression point of view. And to do this, I staged some discrete stages of meristems as we transition from vegetative to reproductive growth. Similarly to what you saw in our sunflower staging earlier, we have a 10-day. At 28 days, we start to see the inflorescence meristem form. There's kind of an expansion of the meristem there. but yet there's no floral primordia. And then finally, around 30, 31 days, each of these little bumps you can see on the outer periphery there are individual floral primordia beginning to kind of bud off the main inflorescence meristem. I added a fifth additional stage that I'm calling the capitulescence meristem, because unlike the cultivated sunflower varieties that we've been using in the lab, lettuce actually produces a inflorescence of inflorescences, or an inflorescence of capitula that I think is pretty unique. So this meristem here leads to this sort of compound structure and will lead to the development of multiple capitula, which is pretty unique from a developmental perspective. In fact, if you look at the next stage here, you can actually see lateral capitulescence branches forming here, which will produce more of those lateral branches bearing more capitula. So after I identified some discrete stages that were interesting to us, we went ahead and isolated mRNA from each of them for sequencing. And we found that looking at using a principal component analysis of gene expression across these stages, we see them bin out discretely. So each of the replicates I collected clustered more closely together than with any other stage. And additionally, you see a sort of natural developmental arc here, ranging from vegetative growth all the way to this final stage where we see floral primordia forming. And then finally, we have some differential gene expression analyses showing that we do have a decent amount of potential gene candidates of interest, making each of these stages unique from one another. And then next, I looked at some specific classes of genes to better understand whether I had accurately captured meaningful stages or not. And as you can see here, we have expression of leafy, this gene I showed earlier, come on earlier than would have been previously expected in the inflorescence meristem here, the capitula meristem. This is consistent with what other researchers have seen in other families, along with two other genes that function alongside leafy coming on in patterns that we would expect. And then next, I also wanted to look at genes that regulate stem cells, specifically in the clavata pathway. And I'll get into this in just a second, but we do see patterns that sort of recapitulate what we've seen in species like sunflower, especially with clavata 3 here. Great. We have a sequence of transcriptomes of each of these stages. And so we've set the stage for more gene discovery, but what does this actual mean for capitulum development? Remember, what's sort of unique to our group is our ability to functionally validate a lot of this stuff using our lettuce model. So let's turn to that clavata signaling pathway I mentioned just a little bit ago. Clavata is a pathway that is highly conserved across all land plants, essentially, and it regulates stem cells in all studied species. Generally, it starts with a small peptide hormone known as clavata 3, which is then perceived by this receptor clavata 1, and it transmits a signal into these cells to restrict meristem expansion. So here you can see to scale comparisons between a wild type Arabidopsis inflorescence meristem, which is relatively small, but if you knock out and prevent clavata 3's function, in the mutants, you get a drastic expansion of the meristem due to a huge increase in the number of stem cells. Interestingly, this pathway exists in a similar manner in both the shoot and the root. There's a lot of parallels in signaling between both of these meristem types. That might seem like an aside, but I'll circle back around to that in just a bit. So given that we have this period of meristem expansion, kind of underlying capitulum formation, one of the questions that might be natural to ask is clavata 3 signaling, or is clavata signaling suppressed during capitulum development? And looking at expression from both lettuce and sunflower, we see that during this period of meristem expansion, we actually see a pretty big uptick in clavata 3 here, leading us to ask, does clavata 3 still restrict stem cell number in the Asteraceae, or has its function kind of shifted? To get to this, we turned to reverse genetics. Before I show the mutant plant, I want to kind of set the stage with wild type lettuce here. So here we have our capitulescence we discussed earlier, our capitulum bearing multiple florets, our individual florets featured here, and then there's stigmata. And importantly, there are always two stigmatal lobes in each of these species, or each of these florets. This is a feature that's conserved across the entirety of the Asteraceae, and we expect it to be pretty developmentally consistent. However, if we look at the clavata 3 mutant that we generated via CRISPR-Cas9, we see that this number of stigmata goes from 2 to 3 or even up to 5 stigmata instead. This increase in organ number is indicative of a classic clavata mutant phenotype. This is because if you increase the number of stem cells within a meristem, it often results in an increase in organ number as well. Additionally, you see an increase in meristem size, similar to like what you see in the inflorescence meristems of Arabidopsis. And you also see this increase in organ number. Yeah. Okay, so to further validate our mutant phenotype, we went ahead and rescued it as well. So we took our clavata 3 mutants and transformed it with the Asteraceae ortholog of clavata 3. And we found that if we expressed the lettuce copy of clavata 3 under the lettuce clavata 3 promoter, sorry, my computer has a mind of its own today. We found that we're able to return this number of stigmata from 3 to 5 back to generally 2, and for the most part rescue the phenotype. However, if we try rescuing the mutant with the Arabidopsis ortholog of clavata 3, we find that it actually has a gain of function phenotype. Instead of these florets producing 2 stigmata, oftentimes they produce 1 or even 0 stigmata. And this suggests that Arabidopsis clavata 3 signaling capacity is increased compared to the Asteraceae ortholog. If we look at the differences between these two small peptide hormones, we find that there are 3 conserved differences in the Asteraceae that likely underlie this difference in signal intensity, encoded within the first 5 amino acids here. And interestingly, if you look across Asteraceae species, this unique first 5 amino acid sequence that apparently leads to a reduction in signaling capacity is conserved across the entire family. So I hope I've done something to convince you that Asteraceae stem cell regulation appears to be fairly unique, at least compared to other model systems studied so far. However, doing these stable knockouts and complementing them and this kind of functional work is pretty time and resource intensive. And so I wanted to know if there's a faster way to explore stem cell signaling dynamics in the Asteraceae other than looking at the shoot. And as I said earlier, similar stem cell regulating genes are also involved in root meristem regulation. And so I think that the root might be key to faster analysis here. So turning to roots a little bit, because clavata signaling controls the number of stem cells in meristems, if you turn up clavata signaling in a root meristem, it essentially reduces the length of the root due to a decrease in the number of stem cells. And so if you grow both Arabidopsis and lettuce seedlings on media containing the Arabidopsis ortholog of clavata 3, you see a significant reduction in root length. However, if you grow either species seedlings on media containing the Asteraceae ortholog of clavata 3, you see they are completely insensitive to the peptide. sort of recapitulating the results that we saw looking at floral organ number. Essentially, that the Arabidopsis ortholog seems to be a stronger signal peptide than the Asteraceae ortholog. However, where things get particularly interesting is when you look at the intermediate versions of each peptide. So in the bluish gray and in the pinkish here, we have versions of these peptide hormones that are sort of Frankensteins between the Asteraceae ortholog and the Arabidopsis ortholog. See a sort of stepwise response where the roots have a sort of intermediate response to these intermediate peptides, whereas in Arabidopsis, you see a more binary response in which it seems like the roots either completely perceive or don't perceive the peptide at all. And this suggests that there are some key differences in the receptor genes expressed in the roots of each of these plants that have a different capacity to perceive each of these peptides. So instead of doing all this work in the shoot, I propose doing this work in roots instead. As I said earlier, we do a lot of our transformation via tissue culture to regenerate transgenic shoots. However, we can completely skip the shoots entirely and instead regenerate transgenic roots. This is a much faster process, and I get much higher turnover time of roots regenerated compared to shoots. And a lot of this work has been sort of led by an undergraduate that I work with, Ben Welsh here. And we found that we can turn around hundreds of roots in less than a month. These roots additionally are more easily imaged. They're a lot easier to dissect. And we can use root elongation assays like you saw earlier to do some functional assays with them as well. And so I have been kind of toying with the idea of using our new reference genome to synthesize CRISPR guide RNAs, but instead of mutating more of these genes within these roots, we can use a deactivated version of the enzyme used to transcriptional effectors to essentially turn these receptors up or turn them down and then challenge them with different peptides to see how they respond. So right now, it's sort of a black box how these receptors are responding to these peptides in a different manner. But if we can transcriptionally modulate their expression patterns in roots and then challenge them with different cleaved peptides, we should get a better understanding of what receptors are responsible for this difference in response. And with that, I would like to just quickly plug some methods. If you liked some of the imaging that you saw in this presentation, like these here, it's from a relatively simple tissue fixation and clearing protocol. And we have written up a methods paper that will hopefully be published in the Capitulum Journal relatively soon. So if you're looking to generate similar images, keep your eyes open for that. And just a quick summary, comparative transcriptomics can help us address the potential evolutionary origins of the capitulum. The Asteraceae seem to have some unique stem cell regulation mechanics that we would like to investigate further, using lettuce as a trackable model system for both functional genetics, but also these heterologous systems like I discussed in the root. And with that, I'd like to thank my lab, as well as our collaborators at the Burke, Mendel, and Nimchuk labs. And I would happily take any questions.
Thank you, Reid. Does anyone have any questions for Reid? You can put the questions in the chat, or you can unmute yourself and ask them right now. I have a quick question to get us started. So does having a discoid versus like a radiate capitulum impact stem cell regulation? Like, do you see any differences between the head type?
Yeah, definitely. I sort of oversimplified things earlier when I said that you see the floral meristems appearing just simply from the outside in. That can be different in heterogamous versus homogamous heads. Like for example, in lettuce, there is a sort of outside to in pattern, but it's much more all at once. Whereas in sunflower, you see a much longer time span as floral meristems initiate from the outside in. And there's some cool work showing that leafy that Gene and I discussed earlier might actually be responsible for a discoid versus a ray floret identity as well.
Any other questions for Reid?
Can I ask a quick question? First of all, great talk, Reid. That was really enjoyable, beautiful pictures and stuff. Um,
Thank you.
and I was wondering, do the capitula all develop in a kind of Fibonacci-like sequence, or is it more like that they develop in whorls, sort of?
Um, I think yes to both. Again, it's really going to depend on the species, but I think that that sort of phylotactic patterning of the Fibonacci spirals seems pretty conserved, but it's not always it's not always very obvious in every species.
Okay, great.
Yeah, yeah. Paula Loma's lab has done some cool work looking at auxin reporters, and they show that sort of phylotactic patterning first occurs with like auxin hormone maxima underlying them.
Cool.
Yeah.
Thanks.
Okay. Thank you so much, Reid. So
Thank you, everyone.
now we will hear a talk from Yannick Woudstra, who will be presenting their work on the urban ecology of dandelions.
Yeah, thanks. Let me just get my screen shared here. Um, are you seeing the presentation or the presenter mode?
Presentation.
Oh, great. Thanks. Yeah, thanks for the introduction and thanks for having me on these TICA talks. It's really great to be surrounded by Asteraceae experts and enthusiasts and from lettuce to dandelions, it's not a very big step taxonomically, at least. But from kind of in terms of topic, it is a giant leap, I would say. So I will talk about urban ecology here, which has been my gateway into kind of full-time Asteraceae research. So I currently work at Stockholm University in Sweden, but this work was mainly done in the Netherlands during a previous postdoc that I did at the Netherlands Institute of Ecology and Naturalis Biodiversity Center. And I got this fascination with dandelions through this urban ecology work, and that has turned out to be both a blessing and a curse, which I will explain further today. But in urban ecology, working with dandelions is really a blessing because these plants are literally indestructible. They grow everywhere, both outside and inside cities. They're clonal, so if you want to do like greenhouse experiments, doing several generations, it's really easy to work with them. So they're kind of like an ideal model plant for studying urban evolution. And so we use dandelions as a model plant, and using that model plant today, I will build kind of a picture of how you build the perfect urban plant, basically. And I also try to convey some love for these generally unloved plants. And the research, started in this very tiny but very densely populated country of the Netherlands, where I'm from. So here you see a picture of Amsterdam with the famous and iconic canals. And in between the tourists, you can find dandelions growing there as well. So normally I don't really go into this because when I say that I work on dandelions, most people, actually even most botanists, just shrug their shoulders and like okay, that's fine. Um, but dandelions in this presentation, I mainly refer to the common dandelion, which is known as Taraxacum officinale mostly, which name in the broad sense kind of refers to everything that falls under Taraxacum section Taraxacum, which you might have known by its previous names, section Ruderalia or section Fulgaria. And this is a huge apomictic complex comprising many sexual diploid plants and even more asexual triploid plants. And so currently there is about 800 microspecies described in this apomictic complex. And that's because it's of the so-called hybrid swarm type. So that means that diploids and triploids co-occur in populations, and they hybridize to make new apomictic lineages. So we have this constant flux of new clonal genotypes arising, and then these clonal genotypes, they can be divergent enough to be recognized as morphologically different and hence described as microspecies. But it also means that diversity, so there's a lot of which is kind of ideal for doing environmental filtering with heavy selection pressures. And perhaps one of the strongest selection pressures that we can think of is urbanization. Cities present huge challenges to plants. They affect all stages of their life cycle. And it's the most consistent pressure in urbanization is urban heat. So cities are much warmer than their rural surroundings, and that's caused by the retention of heat in buildings and roads and other impervious surfaces that have low albedo. And this affects the growth rate, of course, which could actually be a positive effect because if the temperature increases a little bit, then photosynthesis can become more efficient. But that's, of course, not the case when it leads to a big heat wave. But it also kind of interferes with the natural seasonal rhythm that these plants experience. So the timing of reproduction as a result can be quite off. But even if those plants can survive the heat, then they face the humans and their pets that inhabit these urban environments. And we step on plants, we chop them to pieces with our lawnmowers. And so this makes it even more critical for the plants to time their reproduction right. And of course, that also, in a more indirect effect, affects their growth rates. But then in urban environments, we generally also have fewer pollinators. So that makes reproduction a bit trickier. And in fact, the suitable habitats to grow in are usually heavily fragmented. So that means that there are fewer sexual mates around as well in general. So actually, these conditions kind of favor asexual reproduction. But even if plants do manage to reproduce, then it may be more difficult for your seeds to find a suitable spot to grow the offspring. So dispersal is also heavily affected. Didn't want to So we also need to consider that there's a general gradient of decreasing urbanization from and a nearby park can therefore provide a kind of cool oasis for plants where they experience very different conditions. But we also have roadside verges, which are kind of green vegetation strips next to busy roads, which we also find outside of cities, of course, because we also have roads there. But there's generally more green space available. And finally, if we really go outside of cities, of course, we find more green areas like farms and nature reserves. So we try to capture all this variation as much as possible using urban-rural transects. But every city is, of course, slightly different. So we now also try to incorporate different cities of varying sizes in this project. So how is a dandelion impacted by urbanization? Let's take a close look at the life cycle to understand this better. All urban dandelions are asexual, at least the ones here in Northern Europe, but they still need to flower in order to activate their clonal seed reproduction. So all of the stages of the life cycle are still affected. And to flower, the dandelions usually require a very long winter. So typically they need around 3 consecutive months where the temperature drops below 4 degrees Celsius. Seeds develop within 2 to 4 weeks, and then they disperse the seeds by wind. To disperse seeds, of course. So you might say that humans, and especially kids, do quite a good job at dispersing dandelion seeds, of course, even without the wind. But anyway, when a seed lands somewhere, it can germinate immediately. It doesn't need fertilization to start germinating. And then if it lands on a suitable patch of soil, of course, because that is not guaranteed in an urban environment, it then has the whole summer to grow until the fertilization starts to kick in winter. And these plants are perennial, so they can produce they can reproduce several times in their life cycle. So you might think that missing out on one reproductive season due to some reduced fertilization, for example, is not a big deal, but it's a matter of numbers because every dandelion basically produces capitula with up to 200 seeds. And then these plants usually produce several capitula in one growing season. So if you miss out on one reproductive season, then the competitors have an advantage of literally hundreds of seeds. So that definitely impacts selection. So the first on our list of impacts is urban heat. If cities are warmer, as shown here by a heat map of Amsterdam, then that may actually be beneficial for the growth of the plants. At slightly higher temperatures, photosynthesis can become more efficient, given that there is, of course, enough water. But of course, there is a limit, and the urban heat artifact actually increases the frequencies of extreme heat waves as well. So that can be quite disastrous. And outside of the growing season, this urban heat island effect means that winters are generally milder, so that affects the fertilization. We investigated the effect of the urban heat island in a big experiment where we collected 120 different dandelion accessions along this urban-rural transect that I showed earlier in Amsterdam. And we grew them at different temperatures and then measured the biomass after 11 days. Summer temperature in Amsterdam. We don't see any difference between urban and rural plants. They basically do equally well. But then when we increase the temperature, biomass, so they start relatives basically. And we see this at both 26 and 32 degrees. So not huge increases, but we see as the temperature increases that the effects start to also be diminished a little bit the higher we go. But then if we increase the temperature even further to like 38 degrees, then we see that none of the plants are really happy and that the whole urban versus rural effect basically disappears. Uh, uh, but of temperature difference in rural dandelions. So how does it affect the flowering of the plants? Using the same accessions, we apply different winter regimes by kind of shortening the vernalization time that we expose the plants to. So we reduce the number of days that these plants are exposed to 4 degrees Celsius. And in the absence of vernalization, even put them choose any capitula. So it's we don't see any difference between urban and rural plants. That basically very few of the plants actually. But then if we apply a very short winter, so just over a month basically, then almost all the street plants already start to flower. So there we start exposing like differences between urban and rural plants. And then if we increase the temperature, the winter even further to like something that would be more standard for dandelions, we see that literally all the urban plants have basically started flowering. However, in the rural areas, we still find some plants that have not reached a sufficiently long winter. But we see that park plants, for example, they're quite happy with this kind of long winter. And another interesting thing that we found about reproduction is that this is in a different city in Maastricht, a smaller city in the south of the Netherlands. We found that in suburban areas, so outside of the city center, but more like suburban lifestyle with gardens and everything, we found that the plants usually have a very short flowering season. So they start flowering, but then within a few days already they start producing their seeds. So they really shorten this development time of the seeds, basically. And when we checked human activity in these locations, we noticed that there was a lot of mowing because a lot of these plants are growing in parks and heavily mowed roadside verges. So that explains the kind of need for wrapping things up quickly. You know, you flower and then you almost immediately start producing the seeds. So in these locations, mowing was very frequent, and we see that the flowering season is shortened there. So the transition from basically yellow to brown in this graph. And this seems to be inheritable because we found the same differences for the same accessions in the greenhouse. The final thing I will discuss that is affected by urbanization is the dispersal. And dandelions are kind of, the example plant of wind dispersal, I would say. So they disperse their seeds on the wind by this disc of pappus hairs that is attached to the achene. So it floats on the wind, and then, of course, the plants can play with this by increasing the number of hairs, but also the weight of the seed influences how long it can float in the air. So a plant can really steer how far the seed could fly potentially, and therefore it can steer the dispersal distance, which is quite important in urban habitats because, as I show here in a picture, if a plant grows here in a crack of a pavement, then okay, where the mother plant grows, that's kind of a good area because there's soil available there. But everything outside there, it's kind of bad to land on because it's asphalt, so the seeds won't be able to germinate there. So steering where you put your seeds is quite important here. Um, and so when we started studying this in dandelions, we first looked at what other studies have done for this. So there is a very famous one of the first urban plant adaptation studies from 2008 by people in Montpellier was actually on another Asteraceae on Crapis, and they showed that the urban Crapis plants they produced heavier seeds with almost no dispersal mechanism. So basically, the plants just stood there in their good location and they dropped their seeds right next to them. Because was their reasoning, if the mother plant grows in a good location, then that is by definition a good location to germinate in as well for your offspring. So there seems to have been kind of selection for this. So we expected to urban environments with very short cracks of pavement, consistently like very so we were kind of the length of the flowering period. And finally, it seems to be better to disperse your seeds at questions still, of course. So I've planted some of them here. These are kind of things that we're still working on. But yeah, urban environments are really fascinating. This was work done in collaboration with these people, mainly at the Netherlands Institute of Ecology and Naturalis Biodiversity Center, but also some other universities in the Netherlands were involved. And I'm continuing this work at Stockholm University. So this is a great team. This has been a great team to work with. With the dandelions. So, like I said at the beginning, working with dandelions is both a blessing and a curse. And I've shown you the great parts about it in terms of urban ecology, but now let's talk about the curse. So, working with dandelions has made me fascinated with apomixis. And now nearly all of my research is unfortunately heading in that direction. So if I stay with urban ecology for a moment, I'm currently investigating whether asexual reproduction is indeed increasing as I thought it would be in cities, and whether species are switching to more asexual forms of reproduction, or is self-fertilization increasing, and what effect does that have on the genetic diversity of urban plants, right? But then since I got really so obsessed with apomixis, I started to have a million questions about this fascinating form of reproduction. But one that I really could not shake was that I was wondering why these apomictic dandelions still produce so much pollen. Like, it's literally useless to them. They, especially here in Northern Europe, where there's not even sexual diploid plants around to maybe hybridize with. It's completely useless. And in fact, I did find a couple of dandelions here in Stockholm that do not produce any pollen here anymore. So this is now a new project where I will look at the evolution of pollen under asexuality in dandelions, and I will see what changes in the gene expression, if the quality of the pollen decreases, etc. And that has recently been awarded very generous funding from the European Union. And of course, once you work with apomictic complexes, you can't escape the microspecies. So many botanists have now declared me quite insane to want to work with this, but I mean, someone's got to do it, right? So I would like to at least try to answer the question around what microspecies and sections actually represent in Taraxacum. And I will start rather small with this little section called Hamata, which currently only comprises about 30 microspecies. You can actually recognize this section quite easily because it produces very dark involucral bracts on the buds of the capitula. They're almost black, as indicated in this picture here. But sadly, for most people, you can only see these beauties here in Northern Europe. And the very final thing I will talk about is that there's quite a few nice perks in working with urban flora. I find is an extremely useful tool to communicate to the public about plants. So it's really quite rewarding when you can cure someone of plant blindness, you know, when they start seeing plants as actively living things instead of kind of green background that is there to be ignored. And since I arrived in Stockholm a bit more than a year ago, I got involved in this big outreach project with a group of artists and architects. And our project is called the Ruderal Garden. So kind of the disturbed garden, if you will. And we try to show the inhabitants of Swedish cities what kind of beautiful things can start growing if you leave a place or even a heavily urbanized one unattended. So the highlight so far has been the production of a little ruderal garden in the parking spots of the Contemporary Art Museum where we do this project. And there we literally cut open the asphalt, so exposing the soil that was there beneath it. And we just let the plants and the animals come in. So the public came over and they helped us build a garden from recycled materials that we found in the area. And then we just let like nature run its course, basically. And associated to that, we gave some lectures about, especially because, you know, urban environments are a really great place to see Asteraceae. So thanks for listening, and let me know if you have any questions.
Thank Sorry that it was recording while you were in. We'll splice those back together and hopefully we didn't miss too much dandelion awesomeness.
No worries.
Do we have any questions from our audience? So you can throw it in the chat or you can unmute yourself. I have oh, hold on. Yes, thank you. So I guess I applaud you for tackling the Taraxacum. So I don't think you're a cuckoo bananas, as my 8-year-old is now saying, botanist. But I would love to hear, you know, a little bit more about at least those 30 microspecies. Like, what are some of the first methods you're going to approach with those? What's your game plan for tackling that?
So I'm going to do a lot of target capture sequencing. So that's with a tool that I developed for dandelions, and with that I can easily access herbarium material. And this is really essential because if you want to make sure that you're sequencing the right thing, especially with these microspecies that are extremely difficult to identify, right? Like it really requires very niche expertise to be able to say for sure that it's this. So, but luckily there's a lot of like good herbarium collections out there from like Taraxacum nerds that have collected these over the past couple of decades. So we have a lot of very good verified herbarium material. So I can sequence that. And I did some tests with that, and I can with the target capture data, I can easily like pull them apart in clusters and at least see like, okay, do multiple individuals that have been identified as the same microspecies clustered together, does it even make sense like genetically? And then if I can expand that, I hope to kind of verify what belongs to the same section and what not. And then link to that, I would like to link it to like verified pictures and like from the field, but also like scans of herbarium material, and then use a kind of convolutional network approach to so like a neural network approach to do some like artificial intelligence based identification. And hopefully, like ultimately, I would even like to identify that as like an expansion to something like iNaturalist, you know, so that you can like go in the field and take a picture of that dandelion and say, okay, this is not just like Taraxacum aggregate anymore, right? This is now Taraxacum blah 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 in section Hamada. That that's the dream, but
I think that sounds like an excellent use of AI.
yeah, right.
Yeah.
Let's give AI the most difficult
Uh-huh.
questions.
That's right. Comes from plants, comes from Asteraceae.
Yeah.
Let's see. Isaac dropped a question here. Thanks for your great talk. A lot of the characteristics you mentioned that make dandelions a super urban plant might also make them successful in other extreme environments like deserts and islands. Could you comment on that a bit?
Yeah. Um, That's an interesting question. I do not know if dandelions are super common in deserts actually, because I guess there the main thing would be like drought tolerance or desiccation tolerance or like surviving seasonality in in rainfall. And actually, I used to urban environments are very dry, very difficult to access water. But I've learned over these years that it's opposite because if you land in a crack between pavement stones, then of course it might be difficult to access the soil. But these dandelions have really good tap roots, you know, so they once they can crack between those stones and they can access the soil that's underneath that soil is actually quite well protected, so there's not really any like evaporation from the soil moisture. So in a lot of urban places, once you manage to access that soil underneath, there's it's quite well watered. But yeah, in terms of temperature related adaptations, that's true. There are some parallels there. Yeah. Thanks for the question.
Awesome. Okay, well, thank you everyone so much for coming. We have our next TICA talks in April, and we actually have two talks that day, and then also a lightning talk from Isaac, who's asked the last question. And so I hope to see you guys then. And yeah, have a good one.
Thanks, and enjoy your day.
Thanks.
Thank you, Yannick. Bye.
Yes. Bye.
Bye.